Thank you. Hi, everyone. I'm so sorry that I was late. I apologize. Let's see if I can get this up and running now so that you can see it on the full screen. There we go. Okay. All right. So we're lawyers, and most of us became lawyers, of course, at least they say, because we have an aversion to both math and science. So I'm sorry to tell you that unfortunately a lot of what we're going to be doing today is going to involve math and science, which is, the downside. The upside, though, is that as lawyers, I think we have a unique ability to learn, to continuously grow, and to continuously evolve our practices and our knowledge. And that is part of what we're going to focus on today in unraveling the helix and learning to defend, or at least to meet or deal with DNA evidence. So DNA evidence, in terms of its power, I think at this point it's become a household word. Most of us know that it stands for deoxyribonucleic acid, but even if you don't, you know that DNA is useful in determining things like ancestry. So how many people have gone to Ancestry.com or have submitted DNA profiles to try to figure out their backgrounds? So it's used commonly for things like that. It can be used to determine or to cure or to treat certain genetic diseases, and that's one of the huge advances that it has. In terms of the criminal justice system, obviously it's used in exonerations a lot of the time as opposed to in convictions, so it's been an incredibly useful tool from that standpoint too. And of course it's used in criminal trials and in some civil trials to try to establish that a profile that is found from a sample of evidence belongs to a specific individual. And because DNA is so unique that we measure the probabilities for a discrete and valid profile in the quadrillions or quintillions, 1 in a quintillion. When we deal with numbers like that, we have profiles that are so unique that they're often regarded as being infallible. But DNA, like any other science, is not infallible. It is subject to a lot of possible errors, to a lot of possible confusions, to a lot of possible mistakes. And in order to be able to understand it and to effectively use it, we have to start by understanding the science and by understanding what DNA actually is and how it's used. So first, with respect to the power of DNA, the Court of Appeals has recognized that DNA evidence holds a unique status within the criminal justice system, having a capacity to persuade the trier of fact of a defendant's guilt unlike anything known before, and operating to shroud the prosecution's case with a mystical aura of definitiveness that renders it virtually invincible. If any of you have dealt with DNA evidence at a trial, I think you see that once the jury hears it, it's almost like magic. And the problem with magic is that magic can't be countered. Magic can't be effectively met. But science can be, which is why we need to understand it and be able to explain it effectively for what it is. So for the next few slides, we're going to talk about the basics of the science. In other words, what DNA is, what DNA is made up of, and what the science behind DNA is, so that everyone has a sort of fundamental understanding. Now, I don't have to tell you, I think, that in 2 hours' time, and I'm actually only going to use a fraction of that, maybe less than half an hour to go over this portion of it, there's no way that I'm going to explain all of the science behind DNA. But this should be enough to give you a working knowledge of the vocabulary that you'll hear used when you have a DNA expert testifying, and of how DNA actually works, and basically what you're going to hear the experts testify to at trial. So this is the basics of DNA. DNA is deoxyribonucleic acid. It is present in the nucleus of almost all of the cells in our body. Now, the reason that I say almost is that they have to be nucleated cells. So interestingly, one of the things that people don't know or don't realize, one of the cells that does not contain DNA, interestingly, is a blood cell. A red blood cell does not contain DNA because it happens to not be a nucleated cell. So that's just an interesting little piece of information. But almost all of the cells in our body contain DNA. The cells in almost all organisms contain DNA. And the other thing about DNA that's so special is that the same DNA profile, or the same DNA strand, is not only contained in virtually all the cells in our body, but it's the same from cell to cell. So whether you have a skin cell, whether you have blood, it's not in the red blood cells, but it's in the plasma within the blood, whether you have saliva, which has skin cells within it, whether you have a liver cell, whatever you have, it will contain that same DNA profile. So that's an important thing to remember and to understand, because it happens to be one of the limitations of DNA evidence. We'll see later on that one of the things about DNA is, because the same DNA is in every cell, a DNA profile cannot tell you the source, in other words, the type of cell from which it was obtained, whether it was blood, whether it was saliva, whether it was skin, etc. So that's important to understand. Now, in terms of how DNA operates, if it were stretched out, each strand of human DNA would be about 6 feet long and 2 nanometers wide. But in fact, the way the DNA is stored in our body is it's tightly bundled in the chromosomes within the nucleus, and we'll see a picture of those in a little bit. Each chromosome is 1 to 10 micrometers long, and then there are some numbers here that show you the conversions. Now, for purposes of figuring out or understanding how small these numbers actually are, I'll give you a couple of examples. A micrometer is 1/1,000,000th of a meter, and a nanometer is 1/1,000,000,000th of a meter. So in other words, the average ant that you might see crawling around is 5 million nanometers long. So those are the numbers that we're talking about. The width of a human hair is on average 100 micrometers. So when we talk about these sizes of DNA, roughly 50,000 strands of DNA would fit into the width of one human hair. Okay, so that you understand the proportions that we're dealing with. Now, human DNA consists of about 3 billion bases, and 99.9% of them are unique in all people. Only 1%, 0.1% of our genetic makeup, of everyone in this room, of everyone on this planet, of everyone that has ever lived, is going to be unique. So the large majority of it is actually, the vast majority of it is actually the same among all humans. In fact, it might surprise you that among all organic or living organisms, we share a tremendous amount of DNA. So you might have heard we share 50% of our DNA with a banana, right? And that's actually a true statistic. I think some are more bananas than others, so you know that number varies a little bit, but 50% of our DNA on average, depending on the amount of your banana-ness, is shared with a banana. So when we talk about bases, these unique bases, or what makes each of us unique, we're looking at 0.1% of the entire strand that makes up for our various genetic differences among all of humanity. And the bases, just conceptually, are what we will refer to, or what you'll see, constitute the rungs on the ladder. So when you look at a DNA molecule, there's a picture of it here, it kind of looks like a spiral staircase, right? Like a double helix is what it's called. If you were to flatten that helix out to get rid of the twists and turns in it, what you'd be left with is a ladder. So the rungs of the ladder are what we call the bases, and each rung is a base pair. And we'll look at that in a little bit. The order of these bases on the rungs of the ladder is what makes up our uniqueness. So whereas I may have one rung in one place on my ladder, another person will have a different rung, a different color rung, a different sequence rung in that same location. And that's how we get the uniqueness. It's just like we've got a limited number of letters in our alphabet, but we can put them together to get a much larger number of different words. And we've got a limited number of words in our language, albeit a much larger number. We can put those together in different sequences to get a vastly, exponentially larger number of sentences that we can create. And then those sentences can go together to make up vastly different pages, books, plays, etc. Okay? So that's how DNA works as well in terms of how it makes us unique. The information in DNA is stored as a code that's made up of 4 chemical bases. So really all we have that makes up this very complex, very long molecule of DNA is really just these 4 chemicals: adenine, guanine, cytosine, and thymine. And what's unique about DNA is that each of these specific chemical bases pairs with only one other. That's also very important to understand, because that's the way we can replicate DNA so that we wind up with the same DNA in all the cells of our body. And it's also how labs can replicate DNA molecules to wind up with copies of the DNA that might be contained in a single cell. Just one strand of DNA can be copied based on this biological characteristic of the DNA molecule. So the way it works is that A always pairs with T. So adenine always pairs with thymine, and C always pairs with G. So guanine always pairs with cytosine, in order to form units that are called base pairs, which again are the rungs of this ladder. Okay, everyone with me so far? So far, so good? So when the DNA molecule splits, and you see that at the bottom of the page, in other words, when you replicate a cell or when a cell divides, the way that you get exact copies of that cell or exact copies of that DNA is that when you're splitting it, think of what's going to be left on the left side of the rung. On the left side of that broken ladder, you're going to have a sequence that's going to say A, G, C, T, A, whatever the pattern is. Well, the cell knows, because these base pairs only combine in this specific way, everywhere that you've got an A on the left side of the ladder, you're going to have a, everybody, what on the other side? A T on the other side of the ladder. Every time you've got a C on one side of the ladder, you're going to have a G on the other side of the ladder. So that's how it copies. It's designed biologically, beautifully, to be a replicating or copying molecule. And this is the way that it accomplishes it. Now, the labs take advantage of that property of DNA that already exists, and they split it up to create copies in the same manner. And that's how you wind up with multiple copies, we'll see, from a small sample of DNA. Now, each base, in terms of the vocabulary that you'll see, is attached to a sugar molecule and a phosphate molecule, and together they make what's called a nucleotide. So that may be a term that you see. That's nothing more than the base pair combined with that sugar and that phosphate. So that will be a nucleotide. Nucleotides then subsequently get arranged into this double helix, the two long strands that form the double helix. The base pairs, again, are the rungs, and the sugar and phosphate are what wind up being the arms of that ladder, the legs of that ladder, the vertical side pieces. And then, as we said, DNA is designed to replicate when cells divide so that you get this exact copy in the manner that we discussed. Now, within our bodies, DNA doesn't just hang out loose, right? I already mentioned it's bundled into chromosomes, and that's what allows us to have all of this lengthy information, because it would be a very, very, very long strand, right? But it gets bundled up very, very tightly within these chromosomes. So on the right, you have a picture of what the chromosomes in our body might look like within the nucleus, or how they would be represented in a lab. So each cell has 23 chromosome pairs. This may remind you of biology from long, long ago, right? We all learned this. And in other words, 46 total, because they're pairs, with 23 inherited from each parent. So what you see here on the right is you have from 1 to 23, 23 being the XY chromosome that is the marker that determines our gender, our sex, right? So XX for a woman and XY for a male. And so it's 23 chromosomes total. One comes from the mother, the other one comes from the father. And that's how we wind up with these chromosome pairs. Now, you've also heard the term genes, right? Not as in Levi's, but as in DNA genes. So you've heard the term genes. A gene, a lot of people think of it as a unit. Like a lot of people, if I were to ask you, give me your mental image of what a gene is, you would say it's like a little ball, like a little thing that hangs out there somewhere on the DNA molecule. And that's actually not accurate. A gene should be thought of as a location. It's like an address. It's a segment or location of the location on a chromosome that codes or encodes for certain traits. So down below, you'll notice that for those chromosomes, that chromosome pair that you have there, one from the mother, one from the father, there are genes or locations on those chromosomes that correspond. They're both in the same location on each chromosome, and they correspond here on this chart to eye color at the green location, to blood type at the lower location. So those are what we refer to as genes, segments or locations on the chromosomes that encode for certain traits. An allele is one member of the gene pair. And it will be referred to, as you'll see later on, when we look at evidence that's introduced at trial, it'll be a number. We're going to refer to the allele as a number. But the allele is one member of a gene pair. Each human, and you could probably figure this out, because we get one from the mother and one from the father, one gene from the mother, one gene from the father, we are going to have at each one of these locations, or genetic loci is what they're called, locus is the singular, you're going to have a total of 2 alleles, which will be represented as 2 numbers. One comes from the mother, one comes from the father. And we're going to have that throughout our DNA molecule, throughout all of the loci on the DNA molecule. The typical DNA report that's admitted at trial is a list of the alleles that are taken from 13 to 20 loci, or 13 to 20 locations on this molecule, plus the sex chromosome, which will tell us in a DNA report whether the contributor to the sample was a male or a female. Okay, so that is what we're going to see. That wasn't so bad. That was the science of all of DNA that was kind of compiled into 3 little charts. But now what we get into is, so that's kind of the overarching science of what DNA is and the terms that you're going to hear used and how it works. Now we get into the basics of DNA testing before we get into the law. So now we need to know, how do you test? How does the lab actually take this information that we have within our cells and break it down and turn it into nothing more than a chart of numbers that the jury is going to see and be able to compare? So the basics of DNA testing really consists of 6 steps. The first is collection. Collection has to happen simply to get the DNA sample either out of our bodies or out of a puddle of blood on the street or out of, the fingernail scrapings from an individual victim. That needs to be taken from the location where it's found and brought to the lab, right? That is the process of collection. So typically you see on the right a picture of a person wearing gloves collecting a swab of DNA from a stain on the floor that has evidence markers around it. That would be a means by which the area of the substance would be swabbed for DNA. You know that if you've given a DNA sample for purposes of running any kind of genetic profile, or if you've seen a client give a DNA sample, typically they use a buccal swab, so they'll swab the inside of the cheek in order to get a DNA sample, and then that could be brought to a lab. They can take a blood sample that will also generate a DNA profile, again from the plasma within the blood. So all of those are different ways to collect and to then bring the DNA. Now, collection is important to understand because the way the DNA works is, we don't know, remember, we don't know the source of a particular DNA molecule. It could come from a person's blood, it could come from a person's skin, it could come from a person's saliva, right? And it could come from a very, very small amount of any one of these things. So when we move about the world day to day, we're leaving our DNA all over the place. I shook Lauren's hand before, I left my DNA on her hand and she left it on mine. When I touch this desk, I'm going to transfer my DNA and maybe the DNA of anyone I've touched to this surface. So when collection is being done, it has to be done very carefully to avoid contamination, and ideally, collection should also be accompanied, if you have it from an unclean source, not from a buccal swab or from a blood test that's being done, but collection by swabbing the ground. Ideally, as in any science, you want to have a control. So you're going to want to also swab an area of the floor that does not contain the stain, but that's near it, to make sure that the DNA you're picking up is coming from the stain and not coming from the substrate, right? So all of these are things to understand in the means in which the DNA is collected. The second step, third step, fourth step, fifth step all happen once we get to the lab. So the next step is extraction. Now, extraction is accomplished in the lab by the cells that are picked up on the swab or in the sample being broken apart and releasing the DNA from their nuclei. So really what's happening is the nucleus is being broken open. There are chemicals that are used to facilitate that, and then a centrifuge that's used to separate the material out. And then the DNA gets extracted from within the cells, and the other material gets sloughed off in that centrifuge. The next step is quantitation. Quantitation is a step by which a machine in the lab will measure the total amount of DNA in the sample. Now that is very, very important because different DNA test kits require a different amount or a set amount of DNA in order for the sample to be run or for the test to be run reliably. So you need to have a minimum quantity of DNA, and that is measured at the quantitation stage. Now, as we'll see in a little bit, that can be fudged a little bit, and some labs have done that, and we'll look at those testing methods through low copy DNA testing and through high sensitivity testing, where the lab will artificially create a larger quantity of DNA to make up for an insufficient sample. And that's done through the next step, which is also done in general, amplification. When you have a sufficient quantity of DNA to test, and some test kits will say you need 101 picograms, some test kits will say you need 500 picograms, some will say you need 20. So different manufacturers have different protocols where they validated their tests to be reliable. Amplification is the process by which we use that natural process, that natural property of DNA to divide and to replicate and to make exact copies of itself, to create carbon copies. Typically, an amplification of a DNA sample consists of running it through 28 cycles to amplify. Now, you may not think that that sounds like a lot. So we're going to make 28 copies of things and we'll be good. So let me explain to you exactly what that means. The best way to do this is through a fable, which may or may not be true, about the Emperor of China. So it's said that a mathematician brought the game of chess to the Emperor of China. And the Emperor of China played the game, saw the game, loved the game so much, and said, "This is amazing. You are so great. I thank you so much for doing this. I will give you anything you want. You name your price. You can have as much gold as you want, as many jewels as you want, mansions, riches. Tell me what you'd like, and it's yours." And the mathematician said, "You know, I'm a humble man. I don't want to be paid in gold or in jewels. I want to be paid in rice." And the emperor said, "In rice? He's a little mashugana, right? There's something wrong with him." Okay. But the emperor said, "Fine, you want to be paid in rice, be paid in rice." So the mathematician said, "What I'd like you to do is put just 1 grain of rice on the first square of the chessboard, and then double it for each square. So put 1 grain on the first square, 2 grains on the second, 4 grains on the third, then 8, then 16, then 32, until you fill the 64 squares of my chessboard." Now, for those of us who did not recoil from math and didn't become lawyers for that reason, that translates to 2 to the 63rd power. And 2 to the 63rd power, I have to look it up, came out to 9,223,372,364,854,775,808 grains of rice, which was more rice than the entire empire of China had, more rice than the planet Earth has ever and will ever probably produce. Strung end to end, that would be enough rice to get us to Alpha Centauri, which is our nearest neighboring star, and back. Okay? So to give you an idea of the amount of rice that would be, that number, end to end for grains of rice, would span about 60 trillion miles. The distance to our sun is only 93 million miles, and the distance to the moon is only 239,000 miles. So you're talking about a lot of rice. Now, they don't tell us what happened to the mathematician, and I'm guessing he was probably pretty quickly beheaded when the emperor realized that he had been duped. But regardless, that is how amplification works. So when you hear 28 cycles or 28 copies being created, really what that means is with 28 cycles, if you started with only 1 strand of DNA from 1 cell, you would turn that into 268 million copies. Now, has anyone ever made a copy of a copy on a copying machine? Right? So when you make a copy of a copy, what happens to it? It degrades. And if you make a copy of a copy of a copy, it's even worse. And a copy of a copy of a copy of a copy becomes progressively worse. And it keeps going that way. Well, now we're talking to the tune of, we're going to have billions of copies from these 28 cycles. And they're not copies of the original. They're copies of copies. So if there are mistakes that are built in to the copy that's being made, those mistakes will be amplified and will become worse and worse as additional copies are created. That's why when the manufacturer's test kits say you need to have a certain starting quantity of DNA at that quantitation stage, they do that because in order to be deemed reliable, you need to start with a large enough sample that you're sure that the copies won't be copies of one flawed molecule that's being replicated again and again and again. So that's why they run these validation studies and come out with these minimum amounts. Now in low copy number testing, which we'll look at in a little bit, and in high sensitivity testing, instead of running 28 cycles, the labs will run 31 cycles to make up for an insufficient quantity of DNA. But again, it may sound like 3 extra cycles is no big deal, but 3 extra cycles, you're talking about a difference from making 268 million strands of DNA to making more than 2 trillion copies or strands of DNA just by running those additional 3 cycles, like the chessboard, right? It's exponential. And so as a result, amplification is an important stage, and it's important to understand how that works so you can understand why a low quantity of DNA can really be a problem. The next step is capillary electrophoresis. In capillary electrophoresis, the DNA molecules are ultimately separated by size. This sounds complicated. It's really not that complicated. The DNA is positively charged, and what they do is they basically run this solution, this mixture of DNA, through a gel towards a positive anode, right? So just like positive attracts negative in every other way, so too positive attracts negative here. Now the DNA has been tagged in this amplification cycle by mixing in these fluorescent dyes that attach in certain areas. And so what happens in capillary electrophoresis is you have a camera. The larger DNA cells, the larger DNA segments are going to move through the gel, slower than the smaller ones will, right? Which makes sense. So the smaller it is, the faster it moves. And so as a result of that, the camera will basically pick up when these fluorescent markers pass through it. And they will create essentially a chart that I'm going to show you in a little bit that's called an electropherogram. Thank you all for still being with me. We are nearing the end of this portion of it because we're about to look at what the jury is going to see or what you'll see in evidence. So the electropherogram is the chart that is created, and it will be read and interpreted by the DNA analyst. And it looks like this. So each of these peaks, this chart that you're seeing, which is called an electropherogram, is nothing more than a measure of where the camera hits or where the camera reads these relative fluorescent units. So in other words, where during electrophoresis, each one of these tagged markers is passing through the gel and the camera picks it up. And it creates a chart like this. Now this chart actually shows us a number of things. You'll see at the top it shows us the individual adenine, guanine, thymine, cytosine. It shows us all of that. It shows us RFUs, or relative fluorescent units, along the side, which will determine the peak height or the intensity. And it shows us the length of each individual peak along the bottom. That's measured along the bottom. Now this is something of a jumble. So what the analyst is going to do is the analyst is going to set parameters. You notice that on this chart you have peaks of different colors. And you notice that you also have some peaks that are very, very, very small, and then some peaks that are much, much larger. So the analyst has a tremendous amount of power. So when you think of DNA as being just, you just run it through a machine and the machine spits out results, that's really not accurate. The analyst has a tremendous amount of power at this stage too, because he can set the parameters for which of these peaks is going to be counted as being valid data, and which of those peaks is going to be disregarded, either because it's too small and so it's just noise, or because it's stutter, it's 2 peaks close together and they have no meaning, they're just reflections. So as a result of that, the analyst can input parameters into the machine. And what the analyst will wind up with, once that chart that we've seen is separated out, is something that looks like this. Now here we're getting, very close to the DNA profile that the jury is actually going to see. So you'll notice that there are a number of locations that are noted at the top of this chart. So the first one of these locations that I'll look at is D8S1179. That location, or locus is the singular, loci is the plural, is nothing more than an address. It's a particular location on the molecule that we're going to choose to look at. And as I said, each DNA test is going to look at somewhere between 13 and 20 of these individual locations. Nowadays, they tend to use 20 because it tends to be more reliable. The more locations you look at, the more reliable the results. Now, what you'll see here, each peak actually represents an allele. So remember when we talked about alleles, at each location you get one from the mother, you get one from the father, right? So at this location, and I told you the alleles will really ultimately be translated as numbers. That's how we're going to identify them. At each location, you can have alleles that are numbered anywhere from 1 to, say, 45, whatever the numbers are. And they vary at different locations how many permutations or different numbers you can have. Well, at the first location here, this individual has 2 alleles, and it is an 11 and a 13. So his mother contributed one of those, an 11. His father contributed the other, a 13, in whichever order. At the second location, this individual has a 28-29. Now, we know that these alleles are coming from the same individual because, first of all, we have only 2 alleles at each of these locations, and every human being has 2 alleles at each location. That's number one. Number two is, we know that the peak heights of each of these alleles at each of these locations is the same. The peak height really has to do with how much DNA you have. And so, you know, I gave you the example before of I shook Lauren's hand and then I got some of her DNA on my hand. If you swab my hand, you're probably going to get more of my DNA than of the DNA of the people I've touched throughout the day, right? So the peak heights, my DNA, there'll be more of it in the sample that we swab, so I'll have higher peak heights for mine. The other contributor to a mixed profile would have lower peak heights. So I can tell when the peak heights are the same length. That tends to support, it's based on the same quantity, which means it came from the same donor, from the same individual. So here we have a profile. Now there is one peak I want to show you. You'll notice at D7, the third one to the right, D7S820, is really only one peak, but it's twice as high as the others. Sometimes the mother and the father will give you the same number or the same allele at a particular location. In that case, the number will be reported, here I think it's 55 or 66, I can't see on my copy, but it's the same number reported twice. So you still have 2 alleles at that location, one from the mother, one from the father. We know that these are 2 alleles because the quantity of that allele is twice as high, right? The peak is twice as high. If we split it in half, we would have equal quantities of both of those. So this is what we get, or what the analyst creates after he has input the parameters, ignored the stray data, determined what's relevant to look at, what's not relevant to look at, determined the sensitivity of the chart or the test that he wants to look at. Once he's made all of those decisions and input those parameters into the profile, this is the chart we get. And then he goes one last step and he translates it into a chart that shows the profiles. So this is a sample DNA chart that might be admitted into evidence at trial. And you'll see that for each of 15 loci plus the sex chromosome, what you have is the suspect's profile. So in this case, the suspect profile is a known profile. That is the profile that the police obtained by using a search warrant or by taking a buccal swab from the suspect who is on trial, or in a non-criminal case from the donor who has actually contributed his DNA in a known way, so that we know the DNA comes from that individual. So that is the known profile. And that generates this profile. At each loci, you have 2 different numbers at each location. And we know that this particular person is a male because it's the sex chromosome is an XY. Well, now the sample, so we have a sample of DNA that comes from an unknown source. And what we're trying to do is to determine, is that sample a perfect match to the known DNA that we have from the donor? So the electropherograms transfer into this chart, the numbers get copied onto this chart, and what we get is at D8, the suspect's profile is a 1314, and the sample has a 1314, so we would say at that location there's a match. Now, it's not enough to have a match at 1 location, or at 2 locations, or at 8 locations, or at 10 locations. You need to have a match at all of the locations to have a DNA match. The more locations you have, obviously, the stronger it is. Now, let's take a look at some things that result in non-matches or inconclusive matches. So at D7, you'll notice the suspect profile is a 10-12. So he donated, or he contributed 2 alleles at that location, a 10 and a 12. The sample contributed an 8 and a 9 at that same location. So based on that alone, we would go across the chart and say, both alleles are different. So if this was my DNA profile and that was the only difference, I would say, there's no match. You've got the wrong guy, right? Not a match. If you look at D3, the suspect profile was a 16-16 and the sample was a 6-16 at that location. So again, if even one allele is off, you would say it's not a match, right? And those would be sufficient to exclude your suspect from being the donor of the sample, because it simply doesn't match up. It's not exact. At VWA, if you go down the chart to the next red arrow, you'll see that what we have is we have the suspect profile gave a 15-19, or the 2 alleles at that location. The sample was a 15. Now there it's a little bit trickier, right? Because what that tells us is we know that each person has 2 alleles at each location. This is only 1 allele. So what that means is our sample is degraded, either because of time, or because we didn't quite get a good enough mix, or because, you know, the DNA started breaking down over time, so we didn't get an accurate profile. Regardless, it's degraded. When we have something like that, there are a couple of things that a couple of conclusions we can draw from that. I can't say, like I could from looking at D7 or D3, that my suspect profile, my suspect is excluded as a possible contributor, because we know he's got a 15 which matches the suspect's 15, and we don't know what his other allele was, right? It could be anything, and it might even be that 19 that we're looking for. So I wouldn't be able to say that this definitively excludes him as a possible contributor, but I could say that you still can't find a match. So this would be reported as, we cannot exclude him as a possible contributor. Nor, though, can I say that this is an exact match to the profile that I started with. Does that make sense to everybody? And then if you look down the line again, we have a more extreme example of that at FGA, which again is just another address that we're looking at. The suspect had a 24-24, and here the sample has nothing at all, again, because it's a degraded sample. So I wouldn't be able to exclude, however, there would be no match, because you don't have all of those locations showing the same alleles as the suspect profile. Everyone clear on that so far? Okay. All right, so now, here's where it gets a little bit complicated. It hasn't gotten complicated yet. Everyone's sighing, oh no. Okay, so here's where it gets a little bit more complicated. Sometimes you will have a single profile, and everything that we've looked at so far, the electropherograms that we saw, and then subsequently the chart that we looked at, was a single profile. Now I know that that's a single profile that came from one donor, because at each location, everyone, I've only got how many alleles? 2, no more than 2 alleles, right? So I know that came from one person. Sometimes you wind up with mixtures of DNA. And as our tests become more sensitive, we're picking up more and more mixtures, or what some of you may have heard of as touch DNA, which is like the example that I gave you before about touching someone's hand and then you get the DNA, right? So you might have a mixture that is called a deducible mixture or a simple mixture. A simple mixture would be where you have 2, sometimes even 3 individual donors, but we can figure out which donor is which because of the peak heights that we looked at before. So in the example I gave you before, you would expect that more of my DNA would be in a swab from my hand than Lauren's DNA, who I just briefly shook hands with. So you would expect that you would get higher peaks for the alleles from my DNA and lower ones for somebody that I touched. Well, that would translate into what's called a deducible mixture. So it is possible, if you have a donor who at one location gave a 1-3, those are the 2 alleles on the blue triangles, and that's mixed with a 2-4, but you can tell that the peak heights are so different that one must have come from one person, the other must have come from another person, and that is created not just at one location, but at 20 different locations that we're looking at together. Well, then we know we've got two donors. One of them we would call the major contributor, who contributed the larger peaks, most of the DNA, and the other would be the minor contributor, who contributed less. And I can generate two DNA profiles based on that. Now, where it gets a little bit trickier still, is sometimes you'll have what's called a non-deducible mixture. And that is in the yellow triangle example at the bottom. So you'll notice that the alleles that I have at that location are exactly the same as in the prior example. I have a 1, 3, 2, 4, which is exactly what I had with the blue and the red triangles. But with these peaks, all of these peaks are the same height, right? So I can't separate them out to figure out who contributed the 1, who contributed the 3, who contributed the 2, who contributed the 4. I don't know. I know I've got at least 2 people, maybe more, right? Because you have 2 alleles at each location, but I may be missing some, or some may be the same, so I may have a 1-1 at one location and therefore have more donors. So I have at least 2 donors, but I can't tell whether 1 goes with 2, whether 1 goes with 3, whether 1 goes with 4, whether 3 goes with 2. So there are so many permutations that this is a mixture from which no deduction can be made. That's called a non-deducible mixture. This mixture, unlike the one we looked at before, can never, ever provide a match. Okay, does that make sense? Okay. Let me take a look at the, I don't have the time here, but keep track of that a little bit. Okay. So this is a non-deducible mixture. So now, this is the chart that you might see for a non-deducible mixture. So this actually comes from a case that I did that we're going to talk about throughout a little bit more because it shows some interesting examples. But this is a case where what we had is we had a mixture of DNA that was recovered from the heel of a suspect's, from the heel of a victim's sneaker. And the victim had been assaulted and his sneaker had been tossed off during the assault, and he testified that during the assault someone had grabbed his sneaker and had thrown it. And that the person who grabbed his sneaker obviously was therefore one of the assailants who participated in this gang assault. So the victim, what the OCME, the Office of the Chief Medical Examiner, did is they took the sneaker, they found it, and they swabbed the heel of the sneaker. And from that swab, they basically got a mixture of cells of DNA. Now, they ran it through all of those stages, right? So the first thing they did is they extracted it. They did this swab. Now, we can think of some of the problems that come from that extraction, right? So before I do anything else, the program code for those listening at home is 874S8. 874S8. So one of the problems with the extraction, I mean, who can think? Where is this coming from? Yes?
Well, you're walking on a sidewalk, so you're picking up dog poop, you're picking up all kinds of things that are on the sidewalk. Your own, maybe your brother touched your shoes, you know, you're walking around your house.
So the source of this DNA is potentially a problem for the extraction, right? So the extraction stage has some issues. So if this was your case, the first thing you would say, if you're looking at it, is you'd say, I got some problems at the extraction stage. I don't know how accurate this is going to be. If this person walked through, you know, a puddle of spit on the street that my guy had spit out, you know, a couple of days earlier, well, then his DNA would be on it. If he brushed against his DNA or an area that my client had touched earlier, his DNA might be on it. So that's part of the problem with the source of where the sample was drawn from. The next problem here is that the amount of DNA that was recovered from the heel of this sneaker was only 5 cells worth of genetic material in a mixture. So that meant that some amount of DNA must have come from one donor, or well, some amount came from one donor, some amount came from a second donor, and who knows however many other donors' DNA is contained within this exterior of the sneaker. So what the OCME does in that situation is, again, it runs what's called high sensitivity or low copy number testing because it doesn't have a sufficient quantity of genetic material to allow it to satisfy the manufacturer's basic reliability requirements, which for this case were at least 101 picograms, and it didn't have that. So what it does is it runs 31 profiles, 31 amplification cycles instead of that 28, creating more DNA. And then, in order to ensure that the results are more reliable, instead of running that once, it actually runs those repetitions, it runs that DNA test for the sample 3 times. Now, I actually kind of like this because I think it shows you really powerfully, it's a powerful illustration of why low copy number testing, when you're running these additional amplification cycles, you really do get somewhat unreliable results. Look at the repetition from, so the victim's profile is very clean, right? 13, 14, 30, 32. There are 2 alleles at each location clean. The suspect's profile is very clean, 2 alleles at each location. Those were pristine. They were taken with buccal swabs in a lab, clean environment, were good. The first repetition from the sample, from the sneaker, resulted in 5 alleles at the first location at D8, 10, 12, 13, 14, 15. So, we look at those 5 alleles and we can draw something of a conclusion if we were looking at this alone in a vacuum. We would say, how many donors, minimum, must we have? 3, minimum 3 donors, because we've got 5. And if we had only 2 donors, we'd have a maximum of 4 different alleles, right? Minimum 3 donors, and in some combination, but we don't know which, because this is a non-deducible mixture. There's no minor, there's no major, there are no peak height variations. In some combination, they provided these alleles at this location. Okay, well look what happens when they run a second sample. Now mind you, this is the same vial of amplified DNA product. In other words, it's a liquid, right? It's the same vial of liquid that was used to run the first test. Another sample from that same vial is now being taken, the test is run again, and the result at D8 is different. Now we get 10, 12, 13, 14. There's no 15 in that one, right? Because there was such a small quantity of DNA that when that DNA was replicated, even within an individual little vial of liquid, there are variations based on which part of that you take. And so we wind up here with something that might be 2 donors, right? And has a different profile. It's missing that 15. Now it's even better when you look at sample 3. This is the 3rd repetition of the exact same mixture of liquid from the exact same vial generated from the exact same profile, and it's 13, 14. So now this might be just 1 donor, right? And we've lost all of that additional material. So I actually love this because this happens at every single location, and more powerfully than anything I can explain, it shows how much variation there can be when you're using these additional amplification cycles on a very small quantity of DNA. It's very different than the results that you get from a known profile, like from the victim or the suspect, that are perfect and pristine and clean. It's very, very different. So now we have all of these repetitions. So now what the OCME does to make up for that is they say, what we're going to do for reliability purposes is we're going to take a composite profile. So we are only going to consider the alleles that appear in at least 2 of the repetitions at each location as being reliable alleles. The analyst actually testified at trial. The other alleles are fluke alleles. That was his word. They're fluke alleles. They're flukes. We don't know what they are. We can't explain them, so we don't count them. So the analyst created the following composite profile, which you see on the right, in order to count as the DNA mixture. And this was what was reported at trial and what went into evidence. And because it was a non-deducible mixture, they then ran probabilistic software, which we'll look at in a little bit, to determine the probabilities that this mixture in the composite actually contained the suspect or my client's DNA. And they used just the composite profile. Now, one thing that's interesting, and we're going to see it in a bit, in running those probabilistic analyses, which are really statistical analyses, the analyst again has a lot of power, because the analyst gets to draw certain assumptions or conclusions, make assumptions about how many people are involved in this mixture. We know that from Rep 1 and from many of the locations in Rep 2 and in other places of Rep 1, there may have been 3 or more donors here. But based on the composite, the analyst says, I've got a 2-person mixture. And that is the assumption that will now color all of the conclusions that get drawn from this sample. And ultimately, the conclusion that was drawn here, I'll break the suspense. Ultimately, the conclusion that was drawn here was that it was 133 times more likely that my client was a contributor to this mixture than that he was not. And we'll take a look at that more in a little bit. But this is what a profile of a non-deducible mixture might look at. It is a mixture from which no deduction can be made, but statistics can be used to provide meaning to the results in any event. And this is essentially what you might see. Now, that's kind of the science and the way that the lab works in figuring everything out. So now let's take a look at specifics about some of the case law and real application to real cases as to how this actually plays out. So if you are on trial, or if you're dealing with evidence that might be used at a trial that you're involved in, or even if it's your own evidence that you want to introduce, you need to be aware of the following groups or classes of problems for purposes of the validity of the DNA results that you might get. The first one is contamination, and contamination can occur prior to the crime, during collection at the scene, or during testing at the lab, and we'll look at that. The next one is analyst error, which is self-explanatory. The third type of problem can come from the misuse of test kits, and I already alluded to that a little bit in the fact that for low copy number and high sensitivity testing, the manufacturer's protocols about how much quantity of DNA you have to have is actually sometimes ignored, or at least in some programs is ignored, to allow for the same testing from a much smaller product, which creates reliability issues. So misusing test kits, using unreliable or untested probabilistic software, which we'll look at, and then overstating scientific conclusions. That can come from either the expert on the stand implying that he can draw more or a stronger conclusion than what he can actually draw from the evidence that he has, which is something that you need to tackle and expose on cross-examination, or it could come from the prosecutor, for example, in a criminal trial, on summation, overstating the conclusion that can be drawn from the expert's testimony. So there are 2 forms of that. So we're going to look at each of these problems individually with some examples. So the first is contamination. The first type of contamination is contamination before the crime ever even happens. There was a very interesting study in 2015 by the University of Indianapolis on touch DNA, which I've already spoken about, touching a surface, etc. And the example that they did, or what the test that they did, the experiment, was they had person A and person B shaking hands, and then one of them, person A, touched the handle of a knife. Then what they did is they swabbed the handle of that knife to see what DNA they would get from the knife handle. And what they found is that B's DNA, now this is a person who never touched the knife, right? Only shook the hand of person A who touched the knife. So B's DNA was found in 85% of the samples on that knife. So that would be proof at a trial, right? It could be used as evidence to say, B must have handled this knife, the murder weapon, because we found B's DNA on the handle of this knife. But this is a study where the DNA was from a person who never touched it. And then it gets even more interesting, because in 20% of those cases, B's DNA was identified as either the primary or, better yet, the only contributor. In other words, A's DNA, the person who touched the knife, wasn't left on it at all. We're all more familiar with fingerprints, and anyone who's done a trial with fingerprints knows that the experts used to always testify it's a chance impression, right? Sometimes you touch a surface and you leave your fingerprint, sometimes you touch a surface and you don't, right? So if I find your fingerprint on a surface, I know you must have touched it. But if I don't find your fingerprint on a surface, I can't say you didn't touch it, right? That's the way it works. Well, DNA is actually far more insidious than that. DNA is also a chance impression. I could touch a surface and leave my DNA on it, or I could touch a surface and not leave it, right? So when you see my DNA on a surface, you would say, okay, I should say the other way, when you don't find my DNA on a surface, you can't conclude that I didn't touch it, right? It makes sense, because I might have left it, I might not have. Where it's more insidious is that unlike fingerprints, if you find my DNA on a surface, a fingerprint would tell you I must have touched it. There's no other way I could get there. My DNA on a surface does not tell you that I must have touched it. It tells you that it could have gotten there by somebody who came in contact with my DNA, touching it and transferring it on a surface. And there are actually different types of transfer that you all need to be aware of. There's primary transfer, secondary transfer, and tertiary transfer. And it's fascinating how it works. Primary transfer is, I touch this desk, my DNA is on the desk. Primary transfer. Secondary transfer is, I shake Lauren's hand, Lauren touches the desk, my DNA is on the desk. Okay, that's secondary transfer. Tertiary transfer is even worse, because my DNA can wind up on this desk even though I never touched this desk, and even though I never touched a person who touched this desk. So for example, I walk into the Nassau County Bar Association building and I turn the doorknob. The next person who walks into the building, who I don't know, have never met, have never touched, have never spoken to, have never come into contact with, turns the doorknob after me. And then they touch the desk. And guess what? My DNA could be on that desk. So that's tertiary transfer possibility. So that is a form of contamination that can happen before the crime ever happens. And it's very interesting, and it resulted in some interesting results in some cases, some very disturbing results actually, because it can really implicate the wrong person. There's a great case, or a great example, called the Phantom of Heilbronn. And this was the DNA of one woman. She was called the most dangerous woman in Germany, because her DNA was found literally on 6 murder scenes. So they were looking for the profile of this woman, for this genetic profile, who was like this serial killer in Germany. 6 crime scenes, they couldn't link them together, they were murders, they were odd. Well, it turns out that this woman was the most dangerous woman in Germany, was a worker at the cotton swab factory that had been used to swab the samples. And so her DNA transferred on the cotton swabs that were then used by labs. It was distributed to the police in Germany, that they used it for the swabs, and her DNA profile wound up there. So it's a very interesting example of how contamination can happen long before the crime occurs. The next example of contamination is during scene processing. So, for example, you can have the genetic material from first responders at the scene that gets transferred into the items that they're collecting. That happens frequently if you use swabs in multiple areas. So I had a homicide case, for instance, where on cross-examination it was revealed that the police who came to the scene to swab some blood droplets that were at the scene, they didn't change their swab. So they used the same swab at multiple locations. That would allow for transference of genetic material from one location to another location, and it impacts the results. Additionally, if you don't wear gloves, if you don't wear face masks, the DNA of the person collecting it could wind up in there. Failure to change gloves during collection of each sample. Also, another thing that happened in our case, improper handling or packaging of evidence. So we had blood that was found on a rug in a person's home, and there were little teeny tiny blood droplets on this rug. Well, in collecting the rug, they were very careful, and it's funny because the analyst at the lab testified about all his precautions. He disinfected everything. He put a mask on. He put gloves on. He used like 3 different layers of paper. He carefully unwrapped the rug and put it on the table to make sure that there was no contamination of DNA from one area to another. So we asked the cop, not the guy from the lab, but the cop, well, how did you package the rug when you collected it from the scene? And he said, we just like crumpled it up and threw it in a box, right? So that means that if a person walked barefoot over one area of the rug, their DNA could be on that area and could transfer on a stain, which may or may not be a blood stain, that's found on another area. So the packaging or the way that the evidence is collected can result in transference. There's also contamination during testing at the lab, and I think the best example of that, did everyone follow, or did anyone follow the Avery case? Remember Making a Murderer? Did everyone see that? Remember, there was a technician at a lab who contaminated a sample because she must have breathed on the sample, And so one of the samples that was taken was ultimately contaminated because of the lab technician's own DNA finding its way into the sample. So that also can result in contamination. So it's very important that you make sure on cross, when you're analyzing evidence, that the proper protocols for collection and for testing are used to prevent contamination. Now, these are two cases that are actually very interesting, two contamination cases. The first one is People of the State of California versus Lucas Anderson, and this is actually published in Scientific American, and I have the quote here. This is probably, it may actually be the only absolutely confirmed example of transference resulting in charging an innocent man. I mean, not just, it's actually 1,000,000% confirmed and accepted by everyone. What happened here is that there was a homeless man who was charged with the murder of a Silicon Valley millionaire based on DNA that was recovered from the victim's fingernails. The two obviously would have had no contact with each other, had very different walks of life. The problem is that although the DNA sample was gotten from the fingernail scrapings, right, although it was clearly his DNA, the problem was that Anderson, the homeless guy, had an airtight alibi. He was at the hospital nearly comatose with a BAC of 0.4. There was no question. He was out, and he was under constant medical supervision at the time of the crime, and in fact, for a long time before and a long time after. So he couldn't have done it. So everybody said, well, how could it possibly be that his DNA then was found in this area? The explanation, it turns out, was that the paramedics who treated Anderson earlier, in other words, the homeless guy, for when he was intoxicated and had fallen into a near coma, earlier that day they treated him, and those same paramedics then responded to the crime scene. So they inadvertently planted or transferred Anderson's DNA to the crime scene of the victim, who was subsequently found, and I guess to under his fingernails is how it wound up, interestingly. So that is an example of how transferring or contamination can lead to grossly, grossly poor results. Now imagine if Mr. Lucas Anderson had not been comatose in a hospital and didn't have that alibi. There would be no question that he would have been convicted based on this DNA evidence. Yes?
It was only because the doctor who treated him in the emergency room 6 months later saw an article and realized that this was the person that he had treated.
That this was the person. That's how it actually came to light. So it is, I mean, it's really gives you pause. It's very disturbing at how the evidence can really be misused in that way. Another example, now this one is actually gives us a little bit of, the first one made us disturbed, the second one gives us some hope. The appellate divisions have recognized, the courts have recognized this phenomenon of contamination and of transferred DNA, especially touch DNA. So People versus Graham is an example of the third department. There aren't a lot of these cases, but when it's properly argued and preserved, it can actually form the basis for overturning a conviction that has been obtained on these grounds. So in Graham, the defendant's DNA was found on a gun handle. The appellate division reversed the conviction on weight of the evidence review, finding that the DNA could have been deposited on the handle through secondary transfer, and reasoning that the officer touched the defendant when he arrested him and subsequently touched the gun and picked it up to put it into the evidence bag. So this was a reversal. The appellate division said, look, it's not a reversal on legal sufficiency because there was a line of inferences that could have led to this conviction, that could have led the jury to find that the defendant had actually touched the gun. His DNA was on the gun. But the weight of the evidence is against the conviction because there was another explanation that was equally probable that was consistent with innocence. And so this is some of the ways that you can fight this in your cases. And you don't have to wait until the appeal. By all means, you should not wait until the appeal. This should all be exposed at trial to try to preclude evidence in the first instance, and then to try to fight the evidence if it's allowed in. The next type of issue is analyst error, and as we talked about, there are many parameters that are set or defined by the analyst. So in the case that we were talking about before, which was People versus Herskovic, that was the mixed profile from the sneaker that I had shown you. The DNA results, remember, showed 5 or more alleles at multiple locations. What happened there is the analyst ran the data under the assumption that there were only 2 donors. And then he actually made it worse, this part I didn't tell you yet, because while he excluded the peak alleles in drawing the assumption that there were only 2 donors, he then included them all back in running the statistical analysis that ultimately found that my client was a possible contributor. In other words, My client's unique alleles were not found in the reliable composite. Many of his alleles were only present in the flukes that had previously been excluded as unreliable. But all of them were put back in the mix for purposes of running the test. So that is what we should call analyst error, and that because the analyst actually has a tremendous amount of power and authority to make decisions and set parameters throughout the process, it's something you need to be aware of and you need to attack. Another example, in the other case we talked about with the rug, with the homicide case and the blood on the rug, the analyst actually defined the peak heights at the lowest lab limit, which means that there was a very small quantity of DNA. Other labs would have excluded that and said, these aren't peaks from which I could draw a conclusion, but the analyst in that case said, I'm going to redefine the peak heights on the electropherogram to read at a lower number of RFUs, or relative fluorescing units, than what is required. The other thing that can happen is the electropherograms can be read by analysts in a way that is improper. So extra peaks can be discounted by stutter or noise, even though they might be significant. Low peaks that are actually noise and should be discounted may be erroneously counted as part of a profile. And actually, the other thing that really elucidates that is that oftentimes analysts can disagree. So there was a lot of controversy at the OCME. Teresa Carrageen was forced to resign in 2003, for instance, for changing the colleagues' results against protocols. More recently, there was an issue with True Allele, there were a bunch of people that were fired from True Allele, which is one of the programs that's used for fudging certain results on certain reliability studies. So there are all sorts of issues that could result from analyst error, from human error within the way that these otherwise reliable programs are used or misused. Machines or software can also malfunction. So when we talk about DNA, really, we've got the possible contamination at the source, so the sample itself. We then have the issue of what the analyst does with it and how the analyst interprets results. But we also have a computer that ultimately can be drawing conclusions based on a program that was written by other human beings and is drawing information for probability based on data that is contained in certain databases that may or may not be complete or may not be accurate. So you have machines or software malfunctions. A good example of that is the forensic statistical tool that's used by the OCME, which has been criticized lately in a number of places, including in the New York Times. That has been found to drop data from calculations, and there's an admitted 30% margin of error in quantifying DNA in a sample. Validation studies on the forensic statistical tool have shown 163 out of 550,000 non-contributors with a likelihood ratio greater than 1. In other words, out of 550,000 people, there were 163 people who were known non-contributors in the validation tests that nonetheless came out as being likely, having a likelihood probability greater than 1, meaning significant, as being a possible donor. And what's shown is that there was actually 56 with a likelihood ratio greater than 10, 14 with a likelihood ratio greater than 100, 5 with a likelihood ratio greater than 1,000, and there was one poor soul who was a known non-contributor. This was in a validation study, so this was just in testing. We knew he wasn't a contributor to the match, and the FST software nonetheless said that there was a 1 in 10,000 probability that he was actually a donor. So it was 10,000 times more likely that he was a donor to the mixture than that he wasn't, even though we knew he wasn't. So interesting. So there are all sorts of possible errors with that. Now, the case law has also recognized the importance of the analyst to a reliable DNA result. So People versus John is a 2016 Court of Appeals case. This case actually dealt with sort of a Crawford issue, with a confrontation clause issue, where the Court of Appeals held that it wasn't sufficient for the prosecution to put on a random analyst who basically just looked at the results and came in to put in the results as a business record and to say, I've looked at them and I agree with everything that's here. Instead, the Court of Appeals said that because analysts have this important role, the right of confrontation requires that an analyst who did their own independent testing or who looked at the results independently and drew their own conclusions rather than just adopting other people's conclusions, is put on the stand so that they can be cross-examined about all of these possible ways that analysts can manipulate or can misuse or misinterpret the data. So in People versus John, the great quote from the Court of Appeals is, "We will not indulge in the science fiction that DNA evidence is merely machine-generated, a concept that reduces DNA testing to an automated exercise requiring no skill set or application of expertise or judgment. Likewise, the sophisticated software programs require trained analysts who engage in skilled interpretation of the data from the electrophoresis instrument using the computer software with its color images, particularly as to the peaks in the graph to construct the DNA profile." In other words, the analysts matter. You have a right of confrontation because it's important that you understand the science and how it works so that you can effectively cross-examine them and demystify what the jury is going to otherwise look at as just this magic, unassailable result, which it really is not. The next type of misuse is misuse of the test kits against the manufacturer's protocols. So we talked about the fact that high sensitivity testing and low copy number testing, what you wind up with is artificially creating larger quantities of DNA by running these additional amplification cycles. And we looked at some of the problems. So here, the test kit that was used, this is the OCME's low copy number high sensitivity testing that was actually used in the Herskovich case that I'm using as an example. And they've now actually changed, they've discontinued the use of this particular type of testing, as well as the use of the forensic statistical tool. But there are still cases out there that have used these methods that are still coming within the pipeline, that are still available and are still coming up. So it's important to understand this. And the OCME, while they've discontinued it, has not admitted to the unreliability of either of these procedures, right? So it's still important that you be aware that they'll get on the stand and say, yeah, we discontinued it because, we just wanted to make it even better and we wanted 20 locations instead of, but they won't admit to any problems with these things, and the problems existed. So in this case, for instance, the test kit that was used in this particular case required 500 to 1,000 picograms to permit optimal reliable results. However, the manufacturer test kit said that 100, and this was true allele that was being used, 125 picograms was the minimum threshold required to run the test. The sample that they were dealing with had less than the 100 picograms of the quantitation test. And this is actual testimony from the trial, so you can see how this played out. The people's expert said, quote, "You would typically say it's insufficient to continue testing. Because if at the quantitation stage you don't have that minimum threshold, every lab in the country should look at it and say, we don't have a sufficient sample, we can't go on. So we can't tell you anything about it. We're not going to go on past that stage. No profiles generated, no charts, no electrophoresis, no electropherograms, we're done." Instead, the expert explains, OCME, now this is not my words, this was the expert's words at the trial. OCME, quote, "tweaks the protocols by running 3 additional amplification cycles to artificially increase the amount of DNA by creating more copies." And the expert testified as follows. This is the tweaking of the protocols. "Our laboratory looked," now this is the OCME, the Office of the Chief Medical Examiner in New York, "looked and said, we know DNA is there, right? We have to say it's insufficient because no one had ever went into that realm. So we validated high sensitivity testing. That will take us into that lower range of testing. We're using the same instruments. We're using the same kits. We've just tweaked the protocols." And then what this does, this is shown ultimately to produce an increase in inconsistent results when different parts of the samples are tested, which we actually can see just from the chart that they produced, right? From run 1 and run 2 and run 3 being so very different, just from taking different samples of that one little mixture in the vial. So we have that. We have drop-ins, the presence of alleles that shouldn't be there. We have dropouts, the absence of alleles that should be there. And we have stutter or echoes of alleles. So all of these are shown to be effects or impacts of this type of high sensitivity, low copy number testing. This is unsuitable for upload into CODIS, which is the FBI's national database. Also, there was one court, one brave judge, who conducted a Fry hearing in People versus Collins and Peaks, which is one of the cases that I provided for you for your materials. That case is excellent, and it actually provides an overview of how DNA works and of the science and of the problems with low copy number and HST testing and with other testing used by the OCME that is really exceptional. So you should read it if you're interested. But that court found that this does not pass the Fry test. And additionally, there's a front page New York Times article, relatively recent from 2017, that highlighted the Herskovich case among others and showed that this is really a reason that the testing techniques used by New York City, by the OCME, can be tainted and really became tainted. So this is kind of interesting stuff. Nonetheless, most courts have disagreed with Collins and Peaks. It really is an outlier and has been characterized as such. Herskovich raises the issue. I don't know how it will be decided. It was just argued in the appellate division, and we're waiting for the decision, so we'll see how that goes and how far they get into it. Unfortunately, in that case, there was no Fry hearing that was requested. There was no Fry hearing that was held, and so the issue is sort of peripheral. I'll say unpreserved, but sort of peripheral in the court's analysis. So it's unclear how that decision will pan out. But nonetheless, Collins is an outlier. Virtually every other court that has looked at it under Fry has said that it is reliable and it is therefore admissible. But you should still be aware of it to argue it in your cases and to expose it, at least on cross-examination, if the court lets the evidence in, or through your own experts on your case. Another example of misuse of test kits against manufacturers' protocols. There's another case, People versus Oral Nicholas Hillary. Now, this case was a St. Lawrence County case upstate, and what happened here is that the defendant was charged with the murder of a 12-year-old boy based on a mixed profile of DNA that was found under the 12-year-old's fingernails at his autopsy. So initially, what the prosecutors did in that case is they used TrueAllele, which is one of the test kits, and we'll look at some examples of others, as their forensic software tool. TrueAllele came up, the prosecutors got, quote, no statistical support for a match. So using that software, the prosecutors ran it, the lab ran it, they went to a lab, the lab ran it, used this program and said no statistical support for a match between the defendant, Oral Nicholas Hillary, and the DNA from the fingernails. The prosecutors didn't like that answer. So they then went to a New Zealand lab that at the time was using, not quite entirely new, but relatively new, program that they had developed called STR Mix. And when they ran that, that resulted in proffered statistics, proffered at trial, that only 80 people in the country, meaning population of 319 million, would match the profile of the mixed DNA that was recovered from the victim's fingernails, and that Hillary was one of those 80 people. So if you think of the strength of that based on geography, right, what are the odds? He's in the area, he lives in this small town upstate, he knew the victim's mother, and lo and behold, only 80 people in the country, 80 people out of 319 million would have been possible contributors, and he's one of them, right? So the prosecutor tried to introduce this into evidence at trial. Now, what the defense was able to show is that STR Mix was not run at the validated 30 relative fluorescent units that is required. That's the threshold that the manufacturer really optimizes or set as the optimal threshold, which would have, interestingly, if it had been run at that proper threshold, that would have excluded Hillary as a possible donor. Instead, it was run at the 50 RFU threshold, and so as a result, you twist the knobs, you get a different result, which is what they did in this case. I actually think that's backwards. It was not run at the validated 50 RFU threshold, it was run at the lower RFU threshold. And so what happened is they got a different result because they used a different threshold. Now, the case was a little bit more complicated. It had some other problems. They also used random match probability, which had its own issues. So that goes beyond a little bit what we're doing. But the bottom line is, for using test kits against manufacturers' protocols, even a test kit like STR Mix, which is being challenged and is being questioned, but is generally considered admissible and reliable, can be misused if the analyst tweaks the protocols and resets the thresholds that are being used to generate the profile, and dramatically so. So The trial court in that case found, granted the defense's Fry motion excluding the evidence. The decision is here, and really what the court held is STR mix itself is generally accepted, but the manner in which it was used in this case rendered it unreliable and inadmissible. Another example is using unreliable or untested probabilistic software. So STR mix is generally reliable, or has been deemed generally reliable. There are a lot of different types of probabilistic software. So first let me tell you what that means, because I've been referring to it and not telling you. Probabilistic software is used when you have incomplete results due to low quantity or low quality samples. So you're missing alleles at some of the locations. We've seen examples of that. Sometimes you have none, sometimes you just have one instead of two, etc. Or it can be used for complex mixtures of DNA with multiple contributors, so for non-deducible mixtures, where you can't tell who the various contributors are. The programs that exist, there are many of them. There's STR mix and True Allele, which are the most common, DNA View, Lab Retriever, like LTD, LRM Mix, CE-CIT, Euroformix. I haven't seen some of these come up, but I found them in articles when I researched. So there are many different types of programs that can be used. Different programs, as we've seen from Hillary, can generate different results. So True Allele generated different results than STR mix in that case. One said inconclusive, one said only 80 people out of more than 300 million could have contributed, and he's one of those 80, right? So very, very different results. The reason for this is that each program is written by different companies, by different labs, with different protocols, with different algorithms, using different databases. So you wind up with different parameters for peak-height stutter, allele overlap, etc., and therefore different results. Additionally, each one of these is only as reliable as its source code. Source code is what the programmer writes in as the algorithms that the program is going to use to compute these probabilities, right? So the source code ultimately is only as reliable as what's being input into it. The result is only as reliable as what's being input. The data bank information, some data banks are really vast and have a lot of different profiles in them. Some data banks are very limited and are really bad at accounting for things like common races or accounting for things like shared gene pools. So the Herskovich case is interesting because he was a Hasidic Jew, and the gang assault was a group of Hasidic Jews that had participated in this assault of an African American victim. Well, because of the shared ancestry of the particular group of Hasidic Jews in Williamsburg, you're dealing with a really, really problematic gene pool, right? All of the people that the defendant comes in contact with on a daily basis and among whom he lives and who were actual possible assailants were all Hasidic Jews from the same insular community. And the program, the database that was being used, simply had no way and did not account for those types of situations. So there is a report, a 2017 report by the President's Council on Science and Technology, which expressed concerns about the use of these programs beyond the scope of current validation. And actually that report was interesting because the report said, look, this is hopeful and this is great science and could lead to great things, but there need to be validation studies that are conducted to make sure that this is really limited to what it can draw, what it can reliably draw inferences about. And it gave some examples of how these programs, specifically STR mix and True Allele, which are the most common, when they can be reliable and when they exceed their validation parameters. Additionally, another problem with this is reliability often can't be tested because the source code often is not released. It is proprietary software. So the OCME's forensic statistical tool, which is its own type of probabilistic software that it applies to non-deducible mixtures to, ironically, draw deductions from non-deducible mixtures, from mixtures from which no deduction can be drawn, which really bothers me. But the forensic statistical tool, the source code was developed and written by OCME and was not released by OCME to the public. So the defense could not access it to test it to see whether it's reliable or not, and all the validation studies were run in-house. Now what's interesting with that is that recently, relatively recently, a federal court ordered, this is United States versus Johnson, which is noted here, granted a defense motion to order the OCME to reveal the source code of the forensic statistical tool. However, it granted a protective order. So the source code was revealed to the defense expert who was able to look at it. And the defense expert found all sorts of problems in the source code. So unfortunately, we don't have great case law here, which we should have, basically invalidating the whole thing, because what wound up happening is that the prosecution, once that was revealed to the prosecution, they simply withdrew the DNA evidence rather than having the court write a decision about whether that evidence was reliable and admissible under the Daubert standard, which is what this was about. Yes?
That gag order was just recently lifted.
Yes, it was recently listed, and in fact there was litigation for revealing the source code. There was ProPublica and Legal Aid and other people had engaged in litigation to compel the release of the source code. So it's interesting to see what winds up happening with that. Yes?
So if you can't come up with a reliable probability, how does it pass the Daubert test? Because you have to be able to put numbers on it. Even fingerprints would have to pass that kind of a test.
Right, so what winds up happening both under Daubert and Frye is that you have an expert who ultimately testifies to the validation studies that are internally run and to the results of those studies. So for example, in Collins and Peaks, which is the decision that I furnished you that deals with the reliability of both low copy testing and FST, there were various experts that were called for both sides that testified as to the reliability studies, the error margins, etc. Now, in that case, Judge Dwyer said, I don't find that this passes muster, and he held a full Frye hearing. But other cases, there are numerous cases in Queens, in Manhattan, upstate, even in Brooklyn, that have found that it does pass muster, and most of them won't even hold a Frye hearing. They just look at previous decisions that have found that it's reliable. And so, because under Frye, I mean, under Daubert it's really the same thing, but because under Frye you can essentially look at it and say previous courts have ruled that this is sufficiently reliable and generally accepted in the scientific community, we can rely on that too. And Collins, as a result, really has not been followed. So there is evidence that is generated, there are people that testify, there are validation studies that are provided. For FST, for example, the source code was not released until recently, and I don't think that there's been a hearing since that time on it, so I don't believe that there has been. I don't believe that it's been used yet. We'll see what happens with it. Now, some of these other programs have had their own issues. So recently, True Allele had a lot of issues because there were misreportings from analysts about the reliability. So there have been all sorts of problems with some of these various tests. And some of them do release the source code and make it publicly available, others do not. So it really is case by case, test by test, and you have to look at the specifics for each one. FST we talked about quite a bit. This is the forensic statistical tool, which is developed by the OCME to give meaning to non-deducible mixtures from which no match can be made. Now, the first thing I want to underscore is every type of probabilistic software that's being used on a mixture, once you have a non-deducible mixture, I underscored it before, I can't say it enough. The danger of how it's used at trial is it leads the jury to believe that there is scientific proof that is tantamount to something like a match, because the statistics can sometimes sound impressive. But it is not a match, because remember, to have a match, you need to have both alleles at each of the 20 loci that you're looking at be perfectly matched to the sample. And if you don't have that, if you have less than that, or you have a mixed profile, if you have a non-deducible mixture, that is a mixture from which no deduction can be made. So most labs, before they would apply these probabilistic softwares, would say, we can't conclude one way or another whether this individual donor is in this sample or is not in this sample. We don't know. We can't exclude him maybe, or maybe we can exclude him. We can't include him. We don't know. But the statistical software that's being used creates the illusion of a statistic or of meaning. Now, Mark Twain said there are three kinds of lies: lies, damn lies, and statistics, right? That's what all of this is. This is statistics. It is not more than statistics. It is not magic. It is not conclusive. It is not science. It is statistics. And statistics can be manipulated here as they can anywhere else. They can be invalid here as they can in any other form. Lies, damn lies, and statistics. Now, looking at FST, and this actually comes from Collins Peaks, which is great, the likelihood probability, the way it works, is the analyst first defines what probabilities or what likelihoods we're going to compare. So using Herskovich as an example, for instance, what the analyst said is it more likely, well, let me start even more fundamentally. The analyst began by drawing an assumption, which I don't think was necessarily a valid assumption. He assumed that it was a two-person mixture, even though, as we saw, there were some locations in some test runs that had more than 4 alleles, so it must have been more than that, right? He assumed a two-person mixture. Then he said, I'm assuming it's a two-person mixture, so going under that assumption, is it more likely that the mixture contains a mixture of the victim, Taj Patterson, and Mayor Herskovich, or of the victim and some unknown person? The way that the analysis was created, the way that the different probabilities or the different likelihoods were set, was itself flawed. Because a valid question would have been, is it more likely that this mixture contains the victim's profile, Taj Patterson, and Mayor Herskovich, or Taj Patterson and another Hasidic Jew from the same community as Mayor Herskovich from Williamsburg? Because that was the only pool. We didn't have a pool of the entire planet. We had a pool of the Hasidic Jews in Williamsburg, right? So, and there were other errors with the way that the probabilities or the likelihoods were set. So the questions that you ask will impact the results that you get. And that's one of the problems with FST and with some of these other programs. They allow the analyst a tremendous amount of leeway in figuring out how to pose the questions and which questions to pose to get the most meaningful statistical results. Additionally, FST itself is based on only 480 mixtures of DNA. Think of 7 billion people on the planet. We're going to draw conclusions that are going to take us into the quadrillions, into the quintillions, right? And yet, that database contains 480 mixtures obtained from only 61 individual contributors. That is the sum total of the data bank that is relied on by FST, by the Office of the Chief Medical Examiner when it was using this tool in order to assess or to obtain the probabilities that it was going to use before juries to obtain convictions. Additionally, the next program code is 874S8A. Additionally, only 11 of these involved mixtures of the same race, and some races, of course, were underrepresented. And as I said, this failed to pass the Frye test in Collins and Peaks. It was heavily criticized, it was questioning, and now this, as with HST, is currently pending before the appellate division. I will tell you that the oral argument, which you can watch if you're interested, it is online, you can find it on YouTube, you can find it on the appellate division's website, went pretty well, and the appellate division seemed to be very sensitive to the fact that these were statistics, this was not a DNA match, and that this evidence was disturbing, not just because of the types of tests that were used and the manner in which they were done, but also because in the Herskovich case, he was, of course, convicted based on those 5 cells in that inconclusive mixture. But interestingly, the probability that was spit out by the FST, even with all of these problems, was only 1 in 133 times more likely. 1 in 133. So if you knew that you had a chance, a 1 in 133 chance of getting on a plane and dying, you wouldn't get on the plane. But his freedom was taken away for the same probability. And not counting the ability to transfer on a dirty heel of a dirty sneaker, the fact that his DNA could have gotten there in any of a number of ways, and all of the problems with the software and the statistics. All right, so now the next thing is drawing inferences the science doesn't support. And there are a couple of examples. This actually will sort of conclude. There are just a few examples concluding or showing how evidence overall can be misused. So this is my attempt to have all of you kind of apply what we've just talked about to actual cases that you might be confronted with. So this example comes from one of the cases that we discussed, the Harris case. There was testing, presumptive testing of spots on the rug, LMG testing it's called, that presumptively test positive for blood. Now the way that LMG testing works is we don't actually know for a fact that it is blood, we just know that it's consistent with blood. And we don't know, it could be other things, it could be meat drippings, for instance, it's not necessarily human blood, it could be things like certain types of cleaning products can yield a positive result. So it's a presumptive, not a definitive test. So what happens is the police come, they see these stains, they assume that the stains are blood, or they want to assume that the stains are blood. They test them presumptively positive for blood. The next thing that happens is that DNA matching the victim was recovered from these swabs. So the prosecution concluded that the spots must have been the victim's blood. Okay? And that was what was admitted at evidence at trial and used actually at 2 trials to convince him, to convict him. He was ultimately acquitted at his 4th trial. So the evidence here. What is the problem with this evidence? What are some of the issues that we have if we come across this? Why isn't this so persuasive? Anybody? It's late. I'll go through it. The reason that the conclusion is wrong, there are a number of reasons. The first is that DNA, remember, we can't tell what type of cells it comes from, and DNA is species specific. So, DNA, you won't get a DNA profile from anything other than a human with these test kits. So if you had presumptively positive testing for blood, right? But if the blood that you had on the rug was, for example, the blood from a dog, and I say that because interestingly they had a dog who had mange and who would shake and spatter little bits of blood from here and there. Presumptively what would happen then is you would get a positive LMG test, because that's not species specific, and then if the substrate of the rug had skin cells on it, not blood cells on it, when you did the DNA test you would wind up with a human DNA profile, right? But it wouldn't have come from the blood, which is what the prosecution needed to prove and was trying to prove. You can't use the science to show the source of the DNA. And so, again, DNA can come from saliva, skin, blood, semen, sweat, all sorts of sources, and you cannot, by using the combination of these 2 scientific tests, prove that the DNA that you recovered came from a spot of what you presume to be human blood that may not have been on the rug. The analyst was ultimately forced to admit on cross-examination that the stain could have been meat drippings or dog blood, and that the DNA could have been deposited separately from the victim walking on the rug, so it may not have been the victim's DNA. In that case, it was significant because the prosecution was trying to show that the victim was killed at that location, and the defense theory was that the victim was not killed at that location at all, which is what tied the defendant to it. Another example, misuse of the evidence on summation. This is another example from another case, Powell case. The victim was dating the defendant's estranged wife. The defendant claimed that he saw them together, sat and talked to the wife in the kitchen, and then left. The victim was found bludgeoned to death, and the victim's DNA was recovered from a stain on the elbow of defendant's shirt. In summation, the people argued that piece of evidence right there, by itself, tells you he's the killer, because there's no explanation on earth, none, zero, that would explain why the victim's blood is on that shirt. Sounds pretty conclusive. The problem is, the fallacy again, is that you cannot prove that the DNA came from the blood at all. There is the possibility of transference. So just because you have a person who's in the same place as another person when he's alive, they're at the same table, they're touching the same surfaces, he's putting his elbow on the same surface. If skin cells from the victim having touched that surface got on the victim's elbow, and if that presumptively positive but not conclusively proven to be human blood stain wound up having nothing to do with the DNA that was recovered, you would get the same result. So this is an overstating of what can be drawn or what conclusions can be made. It sounds great, but it really is a misstatement of what the science can prove. The other example, we already talked about, the Herskovich case. Now this is interesting because it actually has both LCN and FST used in combination. You have no match here, and yet the prosecutor repeatedly said that 133 times more likely is proof beyond a reasonable doubt, and also relied on additional statistics that said that it was 1 in 695,000 times more likely that Herskovich was one of the people that contributed if you got rid of the victim, right? So we're going to draw an assumption it's a 2-person mixture, and then we're going to assume that the victim's not one of the 2 people, and then we get this really impressive number that we can throw out to the jury. But both conclusions don't work with the proof that you've got. right? Both assumptions. And yet those are the numbers that are used. Even assuming a DNA match, again, the source of the DNA is unknown, could be skin, saliva, etc. The method of deposit is unknown. The victim could have stepped on the sidewalk where the victim had previously walked or spit or whatever, could have stepped in his gum. Secondary and tertiary transfer is possible. So if Herskovich had shaken hands with the assailant, who then touched the sneaker, his DNA would be on the sneaker, and we saw that study. Tertiary transfer. If Herskovich opened a door in his Hasidic community in Williamsburg, and then the assailant later came and opened the same door and then touched the sneaker, Herskovich's DNA could have been on the sneaker. So all of these were admitted by the analyst on the stand. There was no Fry hearing for a number of circumstances, for a number of reasons, but this was all admitted by the analyst on the stand. And in spite of the admission of primary, secondary, tertiary transfer, in spite of the fact that the analyst admitted to that and said he could not tell you, based on this evidence, that Herskovich had ever tied the sneaker, and in spite of the fact that this is the only case that I've been able to find where the only proof against Herskovich was the DNA on that sneaker and nothing else. No confessions, no admissions, no identification, no statements, nothing. He was convicted. So there you have it. This is from the case law. People v. Wright is a great case from the Court of Appeals. There was DNA analysis of a ligature that was found around the victim's hands that showed that defendant could not be excluded as a donor. The prosecutor interspersed references to the ligature with comments about the DNA profile, mischaracterizing the probativeness of the evidence, and the Court of Appeals reversed, holding that the prosecutor presented DNA results, quote, in a manner that was contrary to the evidence and the science, and that defense counsel was ineffective for failing to object. So even though the error was unpreserved, the defense attorney ultimately the combination was found to have violated due process, and the defense attorney was found ineffective for failing to object to the misuse of the scientific evidence in the prosecutor's summation. People v. Rosier is a Fourth Department case. The expert testified the defendant was among 1 in 15 Americans who could not be excluded as a contributor to the DNA recovered from a gun. The DA argued the DNA evidence was overwhelming and posited if the defendant had not possessed the gun, wouldn't science have excluded him? And that was reversed based on the prosecutor's uncorrected exaggeration. So in sum, the trial strategies that I can offer you based on all of this is, 1, don't delay. Get the discovery early, consult with an expert immediately to make sure that it's complete and to highlight and understand problems. You will need an expert to go through the discovery material and all of it. I know we've been very thorough here and everybody is now an expert in this room, but nonetheless, you have to consult with an expert early. Use all available legal challenges. Don't forget to move for a Fry hearing. It doesn't matter whether it's going to be denied or whether it's going to be granted. Even if you know that it will likely be denied, move for it. Don't just challenge the specific test used, challenge the methodology that was employed in your case. That at least will preserve the issue for appeal. Additionally, cross effectively. Use the cross of the expert not only to challenge the procedures, but to highlight to the jury how much how much human error can be involved in the process and the limitations on the conclusions that can be drawn. Have your expert ready, consult throughout the testimony of the prosecution's witness, and then call the defense expert if the cross is not sufficient. And then be very vigilant during summation, not only because you want to make sure that you effectively counter what the prosecution's saying and limit your adversary to their proof, but also because you have to make sure that you're not ultimately deemed ineffective for failing to object, right? And also ask for a mistrial at the conclusion of summation. And so now, relax your backbone, let yourself unwind like this DNA molecule, and good luck to all of you. Thank you very much.